The story of mathematical biology begins in the 13th century with a seemingly simple question about rabbits. Fibonacci asked how a population of rabbits would grow over time if each pair produced offspring at a steady rate. The sequence of numbers that emerged from this thought experiment would become one of the most famous patterns in all of mathematics. What started as an abstraction about breeding animals showed that numbers could capture something essential about how living systems change and expand. Centuries passed before the next major step in the 18th century. Daniel Bernoulli turned his mathematical mind toward a pressing concern of his era. He wanted to understand how smallpox affected human populations by applying mathematical reasoning to disease. Bernoulli demonstrated that numbers could do more than describe nature. They could help predict and perhaps even control the forces that shaped human lives. Thomas Malthus took this line of thinking further in 1789 with his essay on population growth. He argued that populations tend to grow exponentially, doubling and redoubling, while resources can only increase arithmetically. This insight about limits and carrying capacity would later influence Charles Darwin profoundly. But exponential growth cannot continue forever in a finite world. Pierre-François Verhulst recognized this problem. In 1836 developed the logistic growth model, which showed how populations slow their growth as they approach environmental limits. Then came Fritz Müller in 1879. Working in the forests of Brazil, he developed what we now call Müllerian mimicry. His argument was remarkable, not just for its biological insight, but because he used mathematics to demonstrate how natural selection would favor the evolution of similar warning patterns in different species. It was the first mathematical proof in evolutionary ecology by the early 20th century. These scattered efforts were ready to become a unified field. D.R.C. Thompson published his masterwork on growth and form in 1917, showing how physical forces and mathematical principles shape the bodies of living things around the same time. Johannes Reinke in 1901 and Jakob von Uexkülln in 1920 gave this emerging discipline its name. They called it theoretical biology. The field was now ready for founders like Ronald Fisher, Vito Volterra, and Nicholas Reshevsky, who would transform scattered insights into a rigorous science. When we talk about this interdisciplinary field, you will often hear two terms used somewhat interchangeably: mathematical biology and theoretical biology. At first glance, they might seem like different names for the same thing, but there is actually a meaningful distinction worth understanding. Mathematical biology tends to focus on the practical application side. It is about taking existing mathematical tools and methods and applying them directly to biological systems. Think of it as reaching into the well-established toolkit of applied mathematics to solve specific biological problems. Theoretical biology, on the other hand, takes a broader and more foundational view. Here, the emphasis is on developing new theoretical principles that can help us understand biology at a more fundamental level. It asks what kinds of conceptual frameworks might best capture how living systems actually work, and sometimes generates entirely new ideas that did not exist before in practice. Though these two approaches often blur together, if you look at research areas like artificial immune systems or amorphous computation, you will find researchers from both traditions working side by side. The boundaries become less important than the questions being asked. What matters is whether the work helps us understand something real about living systems at its core. This field has a clear and practical purpose. It aims to develop mathematical representations of biological processes using the techniques and tools of applied mathematics. Why does this matter? Because describing systems quantitatively allows for more precise predictions about how those systems will behave. When you can express something in numbers and equations, you can test those predictions rigorously, perhaps even more importantly. Mathematical approaches allow researchers to isolate and analyze features that might not be immediately obvious to someone just observing qualitatively. There are patterns and relationships in biological systems that only reveal themselves when you do the math. Mathematical biophysics sits at a fascinating intersection, serving as a dedicated bridge between the physical sciences and the living world. While mathematical biology broadly encompasses the use of mathematical tools to understand living systems, mathematical biophysics narrows its focus specifically to the physical and physico-chemical mechanisms that make life possible. It asks not just how organisms behave, but how the fundamental laws of physics and chemistry give rise to physiological functions. The field dives deep into questions that might seem almost philosophical until you realize they require precise mathematical answers. How do nerves conduct electrical signals? How do muscles contract at the molecular level? These are not abstract curiosities, but concrete problems with measurable, modelable components. Researchers in this area develop detailed physical models of nerve conduction and muscle mechanics, translating the language of action potentials and molecular motors into equations that can be tested and refined at even smaller scales. Mathematical biophysics ventures into quantum territory. Quantum biophysics and quantum biochemistry explore whether quantum mechanical effects play meaningful roles in biological processes. This is not merely speculative physics applied to biology, but a rigorous investigation into phenomena like electron tunneling in enzymes or quantum coherence in photosynthetic complexes. The field also embraces computational approaches through cellular automata and tessellation models, which provide frameworks for understanding how local interactions between cells give rise to organized patterns and structures for students approaching this discipline at the university level. The curriculum reveals just how much foundational physics underpins biological understanding. Molecular spectroscopy techniques like FT-NMR and EPR allow researchers to probe molecular structures and dynamics. Radiological imaging technologies, including MRI, CT, and PET scans, translate physical principles into medical diagnostics. Non-equilibrium, thermodynamics, and statistical mechanics provide the theoretical backbone for understanding how living systems maintain order against the tide of entropy. These are not separate subjects to be learned in isolation, but interconnected tools that together illuminate the physics of life. Something remarkable happened, starting in the 1960s. The field of mathematical biology began to grow at an unprecedented pace, transforming from a niche pursuit into a central pillar of modern science. This was not just a natural evolution of existing ideas. Instead, four powerful forces converged simultaneously, each one amplifying the effects of the others. The first force was the genomics revolution. Scientists suddenly found themselves drowning in data. Gene sequencing technologies were producing mountains of information about DNA, proteins, and cellular processes. This flood of raw data was essentially meaningless without sophisticated mathematical tools to analyze it. Researchers needed new ways to find patterns hidden within billions of data points. Traditional methods simply could not cope with this scale. At the same time, mathematicians were developing an entirely new framework called chaos theory. This proved to be exactly what biologists needed. Living systems rarely behave in simple linear ways. They are full of feedback loops and interconnected processes that can produce wildly unpredictable outcomes from tiny changes. Chaos theory gave scientists the mathematical language to describe these non-linear mechanisms. Suddenly, the seemingly random fluctuations in heart rhythms, population dynamics, and neural activity could be understood as following discoverable principles rather than being dismissed as noise. The third force was the explosion of computing power. Calculations that would have taken years by hand could now be completed in seconds. This opened doors to entirely new approaches. Scientists could run complex simulations, testing how biological systems might behave under thousands of different conditions. Problems that were previously impossible to solve became routine exercises. Finally, there was a growing recognition that traditional experimentation had serious limitations. Research on humans and animals raised profound ethical concerns. Beyond ethics, there were practical problems with reliability and reproducibility. The solution emerged in the form of in silico experimentation, which means experiments conducted entirely within computers. These virtual experiments allow researchers to test hypotheses and explore biological mechanisms without the ethical complications and costs of traditional laboratory work. Let us look at how mathematical techniques actually get applied to real biological problems. One of the most fundamental examples is the logistic growth model. When populations grow, they do not keep expanding forever. Eventually, they hit limits. Food runs low, space becomes scarce, disease spreads more easily. The logistic model captures this beautifully with a simple differential equation that shows a population growing rapidly at first, then slowing as it approaches some carrying capacity. It is elegant, and it works remarkably well for many real populations. But biology is rarely that simple. Consider how patterns form in developing embryos. Alan Turing, famous for his work on computation, proposed something remarkable in 1952. He suggested that chemicals could react with each other and diffuse through tissue, spontaneously creating patterns. These reaction diffusion models explain how stripes and spots might form on animal coats, or how fingers might emerge from a limb bud. The mathematics involves coupled partial differential equations, and the patterns emerge from instabilities in the solutions. More recently, similar models have been applied to somatogenesis, the process by which segments form along the body axis in vertebrate embryos. Then, there is the question of stability. When researchers model malaria transmission, they want to know whether a disease will persist or die out. Stability analysis reveals the tipping points. Small changes in parameters can push a system from one behavior to another. Bifurcation analysis, borrowed from physics, helps ecologists understand when predator-prey systems might start oscillating, or when disease dynamics might shift from steady states to cycles of course. Many biological systems involve randomness. Stochastic processes become essential when dealing with small populations or molecular scale events. A perturbation in one step of a biochemical pathway can propagate through the whole system, sometimes amplifying, sometimes damping out, and at the molecular level, researchers use probability distributions to recognize patterns in amino acid sequences, identifying protein families from the statistical signatures they leave behind. Today, researchers are pushing the boundaries of mathematical biology into remarkable new territories. Consider the human heart. Its rhythmic beating depends on electrical waves that sweep across cardiac tissue in carefully orchestrated patterns. mathematicians have developed sophisticated models that capture these dynamics, helping doctors understand arrhythmias and design better treatments. The equations describe how ions flow through cell membranes, creating the electrical signals that coordinate each heartbeat. When these signals go awry, the models help pinpoint why. Moving from individual organs to public health, we find mathematical models tackling viral diseases in powerful ways. Hepatitis C presents a fascinating challenge. Researchers use differential equations to model how the virus replicates inside patients, and more importantly, how different treatment schedules affect viral load. These models help determine optimal timing and dosing strategies, potentially saving lives while minimizing side effects. It is a delicate balance between killing the virus and protecting the patient. The reach of these techniques extends far beyond human medicine. Bee colonies worldwide face threats from varroa mites, which carry devastating viruses. Mathematical biologists model the complex dynamics between mites, viruses, and bee populations. These models reveal how infection spreads through a hive and suggest intervention strategies that could protect these crucial pollinators. At the molecular scale, researchers grapple with incomplete data. Nuclear magnetic resonance experiments give us glimpses of protein structures, but the data often has gaps. Mathematical techniques help fill in these missing pieces, estimating the three-dimensional shapes that proteins fold into. Understanding these shapes is essential for drug design and understanding disease mechanisms. Even the brain yields its secrets to mathematical analysis. Deep within the subcortex, circuits maintain homeostasis, keeping our internal environment stable. These circuits generate brain waves that reflect their activity. Mathematical models help neuroscientists understand how these waves arise and what happens when regulatory systems malfunction. What connects all these applications is the power of mathematical thinking to reveal hidden patterns across scales, from molecules to organisms.